for your seminar. I just want to stress a couple of things on this session, because genetics is, I would say it's not an integral part to the module, but there are some aspects of genetics that you need to understand. And this session is very much an introduction to those aspects. It is not a see-all and hear-all session on genetics and the genome and inheritance, because for this module you do not need to understand all about transcription and translation. So some of these slides that we're going to see in this presentation are for information. They are for further reading. Should you feel so inclined this evening, or there's nothing on the TV, to get out the old textbooks and look at genetics, then you can go ahead and do that. But some of the slides, and I'll point them out which ones they are really just for your information only, should you wish to explore. Okay. This slide is very important because this sort of insummativeness, can you put your lunch away? I cannot believe you're eating in the lecture. No pressure there. If I can't eat, then frankly neither can you, do you know? So we have got these specific learning outcomes, okay? And they are very specific because they're to do with applying this knowledge to your job as a nurse. They're not here to make you into geneticists or biologists. But there are certain things you need to understand as a clinician in terms of inheritance, in terms of blood groups, for example, when it comes to transfusions, and in terms of how cells reproduce. So these 5 learning outcomes are, in summation, this session. And this is what you need to understand. So if you leave this session, and I've talked more rubbish than I usually do, which is highly likely, then feel free to take this slide and just try and answer those 5 questions, okay? Because they are the key. Okay, so we'll try and answer them as we go along. So the whole point of genetics, of course, is to look at the science of genes and inheritance, and how what you inherit, some of your mum and some of your dad, and all mammals do the same thing. And we're looking at DNA in terms of what is DNA, and of course it's the biological unit of inheritance. So everything that you inherit from mum and dad comes to you via your DNA from them. There's of course 2 types of reproduction when we're talking of cells. One is sexual reproduction, to do with your genetic reproduction, and there's also to do with asexual reproduction, where all your body cells reproduce and produce daughter cells only. So there's 2 specific types of cell reproduction, and those are the 2 that we're going to focus on today. To start the session, we're going to look at chromosomes and genes in a bit more detail. So there's been lots of, a few years ago now, but there was lots of stories and press about the human genome being mapped. And it does contain an awful lot of genes, and these genes are carried on the chromosomes. And all your chromosomes are arranged in either homologous or heterogeneous pairs. And if you think about homo and hetero, homo are 2 of the same, hetero are 2 different, okay, and this is how we remember them. However, the human karyotype has 46 chromosomes, which we'll see in a minute, and 22 are your autosomes. And you have all your body cells have 1 pair of sex chromosomes, and these determine the gender. And if we look at a diagram of the chromosomes in a bit more detail, you can see there you've got your 22 pairs, and these pairs are the same. And then you have the 1 pair at the bottom right of that slide, which is the sex chromosomes, XY for the male, of course, and XX for the female. So on all your chromosomes, in all your body cells, you have these 23 pairs of chromosomes, and the 23rd pair is always the sex chromosome to determine the sex of that cell, or how it's inherited. Each chromosome is a mixture of DNA, which we'll look at in a second, and also some structural proteins. So I want to look at chromosomes in a little bit more detail and what they do and how they look. On the left-hand side, top left of that slide, you can see a chromosome which is non-replicated. It's a single chromatid. The chromatid is a single, obviously, of the chromosome. And when they reproduce and they become replicated, they are joined in the middle by something called the centromere. And the centromere really is just a constricted region of the chromosome that joins the two chromatids to each other. And it attaches to the spindle fibres in cell replication, in meiosis and mitosis. So during cell division, the centromere is split, with half going to each new daughter cell, and each taking with it the chromosomes and the inherited DNA with that. So you can see how those two chromosomes always start off as non-replicated single, and then in cell replication they meet at the centromere to make a joint chromosome, a replicated chromosome, and on it goes. In terms of chromosomes and structure, in X-ray cross-section a little bit really, you can see here the two chromatids, the two single chromatids, and each chromatid is made up of DNA, and this DNA of course is your inherited component, and the DNA is wrapped around the nuclear, the proteins and the sugar backbone in the middle. So sister chromatids, for cellular replication, are joined, they're always identical, and of course as you can see on this diagram they're joined at the centromere. So these classic example here is the sister chromatids are together, but these two chromatids separate during mitosis and meiosis, which is cross cellular division, to produce daughter chromosomes. Okay, so each chromatid is one copy of a duplicated chromosome, and it joins the other copy by the centromere, and the whole point of joining is for cellular division. So those chromatids contain all the DNA information. They're structured in a certain way, so they always join at the centromere, but the whole point of those chromatids is to take DNA onto the next generation when they meet the other half to their same and produce a new daughter cell. So if we looked at chromosomes and what they are, we can see that they're completely full of something called DNA. So we need to look at what DNA could be. Can I just say, I did change these slides 2 days ago on Blackboard so that yours may be slightly different, but it's still got the same information in. So, DNA. You'll never need to know how to spell it, so don't worry about it, but of course it's another kind of nucleic acid. And your genes are carried on the chromosomes in base pairs of DNA, and they're all to do with lengths of this certain nucleic acid. And it's a code. You think about any kind of code for a safe or a safe deposit box, and that's what DNA is. It contains a code for your inherited proteins. And you know, because you're clever folk, that all proteins in your body are generally used either for structural function or for another kind of genetic function. So the DNA for your renal cells will look a certain way, and the DNA, the way they're structured, for your brain cells will look another certain way. They'll all contain your same gene type, but they can be structured in different ways, the way that helix is arranged. Now, the interesting with DNA, and you can see on the top right of this slide, is it's formed from 4 bases. And these 4 bases will always be known as A, G, T and C, and you don't need to know what those letters stand for, but they always couple up in the same way. So you always get, for example, adenine pairing up with thymine, and guanine, pairing up with cytosine, always the same, and you can see that on the top right of that slide. And they're bonded by the use of hydrogen, and it's always on a sugar phosphate background, so the sugar phosphate holds it all together. The bottom of that slide, you can see how those genes consist of the DNA, which is made up of those 4 different letters, and it's a triplet code. Not to be confused by the triplet code, but I want you to focus on the fact that you've always got A will always go with T and G will always go with C. And each code corresponds to 1 amino acid. And what is an amino acid again? A protein, a baby protein, okay. So it makes sense then that these DNA helixes are made up of lots of hydrogen bases, but they are basically amino acids that come together in a certain way, and that certain way is your genetic blueprint. So each code corresponds to 1 amino acid, and it's those sequence of amino acids in this DNA that will determine its function and how it works. So the genes are basically groups of base pairs that are organised into larger units, as you can see here, Those groups of genes form the largest strands, and they're known as the chromosomes. So all the chromosomes that we've seen, the chromatids and the chromosomes in the cell, are all made up of these structures of DNA. And the DNA contains your genetic blueprint. So all your cells will contain the same DNA, but in slightly different structures, determining their structure and function. Now, for purpose of this module, I don't want you to worry about RNA, tRNA and mRNA, transcription and translation. This whole point of protein synthesis on this slide is to talk about how we get to become DNA, how we make DNA. I put this slide in for information only, okay? It is not part of this module. But I don't want you to think about, well, she never told me how DNA was made. That's how DNA is made. Basically, you've got your different types of RNA, which are single-stranded, which come together with some enzymes, and ATP, which is energy currency of the cell, to make up the DNA. But it still uses the same genetic codes of A, T, C, and G. If anybody wants to go into protein synthesis, there's some good stuff in Ross and Wilson, and there's some good stuff in Tortora. But as I said, for this module, we don't need to go into protein synthesis in this degree, so that's fine. It's fine by me, I presume it's fine by you. Okay. So we've looked at what a gene is, groups of base pairs, and we've looked at what chromosomes are and what their centromere is. So the centromere is where the chromosomes come together and join. And of course, the chromatid is the single chromosome which pairs with its sister to make a daughter cell, for cell replication. Now, there's an awful lot of information out there in terms of cell division. I know this goes on to a certain degree about what Paul explained to you in the first week. But one of the learning outcomes we do need to understand for this module is how cells reproduce. How that genetic information is passed on from cell to daughter cell. And there are two types of cellular division for the purpose of this module. And one is mitosis and one is meiosis. Okay, so easily confused between the two. The quick way to remember this is if you think of Dolly the sheep and cloning, then that's what happened in mitosis. It was asexual reproduction. And in your body and in my body generally, we use mitosis to repair tissues and cells that have become worn out or damaged in disease. Okay. So mitosis is like asexual reproduction. It's what your cells do to produce new daughter cells to repair and replace, that whole recycling thing. Meiosis is the opposite of that. It obviously occurs only with the production of gametes, which are your sex cells, so the sperm and the ovum. And these are produced obviously by organisms that reproduce sexually, for all mammals. So two key points there. What is mitosis and what is meiosis? And there's generally always a question in the exam on one of the two. What is it? So always try and remember what one of the two are. So we'll start with mitosis, which is cell-to-cell cloning. Really. So this cloning and production of daughter cells happens, of course, all around the body, and it involves just one round of division, which we'll look at in a second. And we use mitosis to repair when we're growing and to replace damaged cells when they become worn out. And mitosis happens everywhere, of course, under no control of your cells. But it includes one round of division and takes place in somatic cells, and all body cells are somatic cells. So because they're producing asexually and because they're producing with one round of division, the cells that are produced with mitosis, the daughter cells, are always identical in chromosome number to the parent cell. There's never any chromosome or DNA difference. They are simply replicating to make new of the same. And of course, this works. If you think back to the sensibility of this particular part of bioscience, you don't want a renal cell making another renal cell that looks like a brain cell. Do you, really? You want the renal cells to look and feel like renal cells, and you want the cells in your skin to look and feel like skin cells, which is why mitosis is what your body uses to make all your body cells when they get worn out and damaged. There are obviously phases to this mitosis. I must stress, however, that for the purpose of this module, we don't need to understand or know those 5 phases, bizarrely, but they're there if you need to want to know what they are. I think it's important to stress, though, that any cell, when it starts to divide and replicate, does certain things, and they all do the same thing. And that number 1 on the top right there is the cell at rest, as it were. And number 2, prophase, is where the cell starts to behave in a way ready for replication. And you can see on certainly the number 3 and number 4 how the chromosomes are pulled to either side of the cell, ready for full replication. So, in terms of this module, we don't need to understand those 5 phases, but those 5 phases are there should you want to look at them. The other type of cell reproduction we need to understand is meiosis. Now, mitosis happens in all somatic cells. Somatic means body, whereas meiosis happens obviously in the gametes or the sex cells. Okay? Now, whereas mitosis was just 1 round of cell division, in meiosis we have 2. And I guess meiosis is where the gamble lies when anyone goes, when you meet the male and the female sex cells together, in terms of what comes out of that, because it's a complete gamble as to how that reproduction will take place in terms of genetic rebalancing. Where we look at meiosis, we look at halving the number of chromosomes, because we have 2 cells coming together to produce the full complement. Okay. So it always occurs in the diploid cells. So we've got the egg cells and the sperm cells, which of course leads to the formation of the male and female gametes. So all male and female gametes contain half the number of chromosomes, so we call it that haploid. There's another word we need to understand, haploid and diploid. So haploid is half the number of chromosomes, i.e. 23, and diploid, of course, is a full complement of 46. So because we're having sexual reproduction and we're having 2 separate chromosomes coming together, it makes sense then that the egg and the sperm contain half each, and they come together to produce a diploid complemented cell. And of course there's a diagram for that as well, and there are 2 stages to meiosis in terms of sexual reproduction. The prophase and the anaphase, again, they do sit in similar ways to mitosis, but whereas in mitosis it's very programmed and it happens in a certain way, in sexual reproduction, in meiosis, it's a complete random pulling apart of the chromosomes, so determining genetic makeup. So really it's very random, and these two chromatids that pull apart, therefore, are different, and they don't produce daughter cells. It's a complete gamble as to what cell they will produce. But there are 2 phases to meiosis, and I hate to say it, but just like with mitosis, for the purpose of the module, we don't need to know these different stages. So, key slide to sum up the differences between mitosis and meiosis. Mitosis is cloning, for want of a better word, so it's all to do with consistency and producing daughter cells. It's still produced as a 46, so a full complemented chromosome number, and it occurs in all body cells and all somatic cells. And the whole purpose of mitosis, of course, is growth and repair. Meiosis, or sexual reproduction, rather than producing 2 identical daughter cells, this results in 4 different daughter cells, which is why we get the random genetic makeup at reproduction. This occurs only in germ cells, so your sperm cells and your ovum cells of the gametes. And of course, the whole purpose of this is to produce the sperm and the ova for sexual reproduction. Don't forget that in terms of mitosis, there is 1 round of division, but in terms of meiosis, there are 2, which is why you end up with 4 different cells rather than just 2 daughter cells. So in terms of cellular division, that slide is it, which seems not an awful lot really, but I'm sure you're as relieved about that as I am. But in terms of cell division, you can explore in terms of the phases and the anaphase and the prophase, should you want to. You have all the information you need on the slide before that. But again, for the purpose of the module, I'm afraid we don't need to go through those different phases. I can hear a collective yes at that point. That's a good thing. Okay, so the whole point of this genetic nonsense then, when it gets quite interesting of course, when you're trying to work out if you belong to mum and dad or mum and the milkman. And of course it's always quite interesting, and there's lots of talk and there's lots of research into genetic diversity and genetic inheritance and how we've come to be these people that we are. And so what I wanted to do was spend a little bit of time looking at inheritance of traits and how certain traits you can see following the blue eyes, the brown hair, the forearms, that kind of thing can pass through families. Anyone know from your pre-reading what an allele is? It is, thank you. So, you know what a gene is, it's your genetic blueprint, it's a selection of DNA ready to make into a new you. But an allele is alternative forms of the same gene, and I think the whole point of genes and alleles come together when we look at blood grouping. And if you remember when you did blood grouping with, was that with me, possibly? And we talked about antigen and antibody, and you have antigens for things you do have, and you have antibodies for things that you don't. And in a similar way to genes and alleles, so we're going to look at that in a bit more detail. So meiotic, is that cloning or sexual? Meiotic. So we've got this diversity occurs due to in the very first division. So we've got the crossing over of these two non-sister chromatids, so they're half of a chromosome, don't forget, and this exchange of genes. And it's this random alignment that causes this whole diversity when you come into sexual reproduction. So the offspring at the bottom of that slide obviously contain genes from mum and dad, but in a completely random alignment, because you have those crossing over at reproductive stage. So when you're looking at inheritance of traits, if you think about it, you know you've got one set of genes from mum and one set of genes from dad, stroke milkman, okay? So you've got one copy from mum and one copy from dad. So you have these two copies that come together in the chromosome, two chromatids, one from mum, one from dad, come together in the chromosome, and they produce these pairs, okay? So your traits are determined by these pairs of genes called the alleles. And so you've got one pair that's found on one chromosome, and the other is its equal partner. Okay. So don't forget that alleles are alternative forms of the same gene, and they have the same genetic locus. They contain the same genes, but the DNA that they contain could be slightly different in how it's coded. So those four letters, the way they're structured, could be slightly different on each chromatid. Which is why you have a complete random alignment when you're looking at sexual reproduction, rather than you have a guaranteed cloning when you're looking at asexual reproduction. Now, in terms of genes then, I'm not done in variety, but this is another key slide, so please highlight this slide as well, because it contains some key definitions. So you've heard of genes in terms of dominant, recessive and codominant. I think it makes quite sense if you look at it that a dominant gene is a dominant gene. So it's a stronger gene type, and it will mask a recessive gene and get its own way. So you only need one copy of a dominant gene for that trait to show in a subsequent generation. So if mum has a dominant gene and dad doesn't, that dominant gene will show up in the successive generations. It's a dominant gene. It will overshadow a weaker or a recessive gene. So a recessive gene, it's obviously, or it says on the tin, it's a weak form of gene type, and if you're going to have dad who's recessive and mum who's recessive, that will come down to the offspring as a carrier, because it's not a dominant gene. And an example of that, I would say, is something, for example, using a disease like cystic fibrosis, which is a an awful disease of the whole body, but mainly is symptomatic in the lungs and the heart. But if mum and dad both have a recessive CF gene, that means the offspring will not inherit CF, but they will be a carrier for CF. Okay. If mum or dad had a dominant CF gene, then the offspring would have CF. It's a dominant gene. But if both mum and dad have a recessive CF gene, the offspring will not have CF, but they could be a carrier for CF. Codominant gene, in comparison, of course, is, as it said on the tin, really, that neither gene is stronger than the other. And when we're talking about terminology of our genes, we're looking about genotypes and phenotypes. And we talked about this in blood groups as well, which is why you've got that as homework to do a little bit. But genotype represents the genes that you carry or you possess. So that comes from mum and dad. Your mum and dad determine your genotype. But the phenotype is the traits you exhibit. So it's what we see as visible, okay? And there's a way of explaining that if we look at the inheritance, for example, of eye colour. This is called a Punnett square, by the way, but it's a very simple way of mapping out inheritant recessive or dominant genes. So, of course, a capital B is a dominant gene, a baby G is a recessive. So dad's genes are dominant B, recessive G. Mum's genes are both recessive Gs in this tape. So mum's eyes are green and dad's eyes are brown. So you can see easily to plot the UoL axis really in terms of there therefore is a complete one-to-one ratio of the child that couple make of having green eyes. Or half the chance they'll have green eyes, half the chance they'll have brown eyes. Because B is a dominant gene and G is a recessive form of that gene. So you can see how mum has inherited a recessive green and a recessive green from her mum and dad. The dad has a dominant brown from one of his parents and a recessive green from one of his parents, which is why his eyes are brown, because the dominant gene is brown. So bring those two gene pools together and you have a 50-50 chance of any child having either brown or green eyes. If we look at that doing it yourself, for example, on this top slide here, dad's got two dominant brown genes for his eye colour, and mum has two recessive blue genes for her eye colour. So what do you think the outcome will be? All I can do is they'll all have brown eyes. Yeah, of course they will. All eyes will be brown. Because the father's genotype is BB, so that's a genetic prototype, is two dominant brown genes. So his phenotype, or what he exhibits, will be brown eyes. Okay. Mother's genotype, what she inherited, is BB, so what she exhibits is blue eyes. So that all offspring will inherit B from dad, big B from dad, baby B from mum, which of course will give them the genotype of BB, which will be brown eyes. So don't forget, the genotype is the genetic makeup of a cell, and the phenotype is the observable characteristics that you see from that genotype. So genotype you can't alter, it's what you've inherited, but the phenotype is what you show, or what you exhibit. And another good example of using dominance of genes is looking at the blood system. Okay. So the alleles we inherit are one of three, A, B or O, and we did this a few weeks ago, so I'm not going to sort of spend a lot of time on it. But if a co-dominant gene is visible, it has no dominance over anything else. Okay, it's not a dominant gene, it's not a recessive gene, it's a co-dominant, so it has no real exertive value. It doesn't tell something else what to do. So in terms of your ABO system, the blood you inherit from your parents, what we've done with this slide is don't forget the alleles, the alternative forms of the same gene. Okay, so parents are A, B or O, any which one, and you will inherit one gene from mum and one gene from dad stroke milkman, which gives you your blood type, your blood group, okay? And we did this a few weeks ago, didn't we? So you can see, because they're co-dominant, neither is stronger than the other. So because of that, they come together as a balance. So rather than one being dominant and one being recessive, they're co-dominant. And this always happens with inheritance of blood groups. Okay, so dad's push is no stronger than mum's push in terms of that allele. They come together and they're co-dominant. In a similar way, when we're looking at gender determination of offspring, so another Punnett square here. Now, if we're looking at gender determination, is this meiosis or mitosis? Okay, thank you. So meiosis is sexual reproduction. So we've got dad at the top, mother on the left of that axis. And again, similar to the eye colour, dad's chromosomes are always classed as XY, mum's are always as XX. So again, same as the eye colour, this child has a 1 in 4 chance, or a 50-50 chance of being male or being female, okay? And this is always the same. And I think there's a controversial area of research in terms of gender determination, but in regular couples who have regular sex to reproduce, it's a 1 in 2 chance of having a boy or a girl. So, gender is determined by the inheritance of a paired combination of the X and the Y chromosomes. And don't forget, the X and the Y is the father's, the male chromosomes, and the double X is the female chromosomes. So half of a chromosome is a chromatid, okay, and they're joined by something called the centromere, which is the middle point of that. So, inheritance of genetic material between body cells in terms of repair and replication is always mitosis, and the genetic inheritance between people is always called meiosis, okay? So we use mitosis or cloning to ensure that all your body cells replicate in the same way in a preordained fashion. Where this becomes really interesting is when you look at the process of disease, and one of the sessions we'll have next year for those of our adult branch is I do a very long, very boring session on how cancer cells develop, and we look at this in terms of how does healthy cell reproduction become a cancerous cell, and it takes several stages, but that's where I think this kind of subject becomes a little bit more interesting, because this session is very black and white. There's been some very key points. So looking at what mitosis is and what meiosis is, and what's the point of these two different ways of cellular reproduction. Genes can be either dominant, recessive, or co-dominant. And again, when you're doing your reading, look at that in terms of disease process and inheritance of disease. And it's not just obviously CF, there's a whole raft of diseases out there that are inherited, and look at how that dominant process works. And gender is always determined by the inheritance of the paired X and Y chromosome. Now, on your blackboard for today's session, I have put this sheet because obviously there is a lot of termination. There's a lot of terms in genetics, and some can be a little bit confusing. And I've tried to keep this session as simple as I can, because what I don't, and I know I've spoken to quite a lot of you this morning who have already got your knickers in a twist over genetics, which is easy to do, but we're not trying to look at genetics in its totality here. We're looking at it as a means to thinking about cellular reproduction and inheritance, because as clinicians that's what you're looking at, okay? So use this terminology session. When it comes to revision for that little test in May, okay, if you can familiarise yourself with what these words mean, then you'll be absolutely fine. Alright? We've actually managed to finish a lecture, which I think is the first one. Okay, thank you very much.
Unless there's...
You will never need to spell a word.
I don't know how many
Is that the right answer?
It was just I wanted it clarifying because at the beginning Nikki said multiple choice, but Ruth sort of implicated today that somehow there may be some written questions.
There won't.
It's not been written out yet or something.
No, it has. It's solved on it. It's written. There was no written questions. It's all multiple choice.
Well,
I need to make that
we will
play.
do writing exams.
Not until the second year.
Right.
Because it's a level 5 module. This is just level 4.
Yeah.
So actually, so much I can ask you to do. If that makes sense. And I think, as lovely as it would be doing some written questions, because that's where you can really
Because
this module has to be very narrow in a way, because it's like, it'd be nice to go into more detail and stuff and make more sense really, but we can't. We only have so much time and we only have so many weeks as a level 4, and we only have to learn so much. But when we come into next year, level 5, with Paco with me, there'll be a lot more free reign, and there will be written questions in that level.
Because obviously I'm still taking more in, but last year we did the access course.
Yeah.
So a lot of what we've covered so far we've covered on the access course. So in a way it's revision.
Yes, and cover it into more detail.
Yeah.
Here. And I guess that, well, I'm sorry about it. I think some like these, we've got a lot of detail on it. But in the past, that's 95%. I'm trying to do it with confidence that the minute it gets too heavy, you can see it go. And then the movement drops, and then it becomes really hard. So I'm not. This is the same module I taught in Oxford. Like it's a special module, but there's a lot less people. Previously, I think they were. They just did what they wanted.
Right.
And now we've had to come into 9, how it should be. Level 4 first year, level 5 second year,
So gradually sort of break it
so there'll be a lot, there'll be a lot more
break
freedom in the second year to explore certain areas and do different things, and that's where it gets interesting. And this is just the building bricks of it all, which is why I'm trying to keep it as simple as possible.
So it's not wrong of me to sort of put what you're teaching us now into perspective of what we learnt in the access course
No, not
last
at
year.
all. And you've done the access.
All I thought was just need to keep having children.
Yes.
I'm studying now.
Yeah.
Cheers.
Okay. Total learning outcomes is 6. Okay, thank you.
Are you in the H yet?
Yeah, I thought that. I'm taking up the students of God, so I'm
Scarf down there. MC, it's in the, hang on a second, it's in the building next door. Is that
It's ground floor, I think.
If it's 1, it should be on the first one.
I had it written down on a piece of paper, which I now can't find.
Little scarf.
I know. No, it's gone. Oh no, it's not gone. MC 0019.
So it's on the ground floor.
Ground floor that night, I think we should walk in.
You know the dominant gene? You know when you said the father had the dominant gene for brown eyes?
Only in that example, yeah.
Would that mean every single child then would have the brown eyes? Or
Between that two couples.
yeah, could have that two.
Yeah.
Have two
If it went over the side with the barmaid down the pub, it possibly didn't.
No, I'm saying like if that couple had other children, they have brown eyes the same as
As long as those two phenotypes came together to produce that child, they would
that
have the
dominant
same.
gene would come over. Right, got
Yeah.
you.
That's
Double checking.
okay.